Good afternoon, everyone, and welcome to our Thursday afternoon webinar, a study of the clinical and genetic risk factors of cholangiocarcinoma. CCF has been a proud supporter and funder of the GWAS Genomic-Wide Association study for the past 10 years and believes strongly in the research being done. I'm Melinda Bikini, Chief Patient Officer with the Foundation, and I'm also a 14-year survivor of stage 4 cholangiocarcinoma. We're so happy to have Caitlin Van Lith join us today from the Mayo Clinic. Caitlin is a student, an MD PhD student at the Mayo Clinic working under Dr. Lewis Roberts. Her main focus is on the risk factors of cholangiocarcinoma, looking both at epidemiological and genetic risks. Because cholangiocarcinoma is rare, she's working to organize an international consortium in order to band together the resources of cancer care experts globally. She plans to continue her career in the field of genetic epidemiology, and we will have plenty of time for questions and answers at the end of her presentation. So be sure to put your questions in the chat or the Q&A box at any time during the presentation. Thank you, Caitlin, for joining us today, and I'll turn it over to you for your presentation.
Thank you, Melinda. And thank you all for joining this afternoon. I'm excited to share some of the work I've been working on for the last 3 years. So my name is Caitlin Van Lith. As Melinda said, I'm a MD PhD student. So that's a 8 to 9 year program that you'd get both a medical degree and a PhD. So I did 2 years of medical school, and now I'm in my PhD phase. And the work I'm presenting today is what I'm doing my thesis on. So as an overview, today we're going to review cholangiocarcinoma. We're going to talk about what a genome-wide association study is, GWAS. We're going to talk about what we currently know about the genetics of cholangiocarcinoma, and then we're going to go into that project where we're going to hopefully expand a lot on what we know about the genetics of cholangiocarcinoma. So looking first at reviewing cholangiocarcinoma. So as I'm sure all of you know, cholangiocarcinoma is a cancer of the bile ducts. It presents in different locations within the bile ducts, and we call it different things based on where it's located. We have intrahepatic cholangiocarcinoma and extrahepatic. Are you able to see my mouse? Yes, perfect. Okay, so intrahepatic cholangiocarcinoma and then perihilar and distal cholangiocarcinoma are considered extrahepatic cholangiocarcinoma. There are some well-recognized risk factors for CCA, including choledochal cyst, biliary tract stones, primary sclerosing cholangitis is a huge one, and liver fluke infections, which are more prevalent in certain areas of the world like Southeast Asia. However, most cholangiocarcinoma cases don't have any of these known risk factors, and most people with those risk factors don't end up developing cholangiocarcinoma. So there isn't a clear association between why cholangiocarcinoma arises in people. And it is difficult to diagnose this cancer early, and so prevention and screening are really key in our ability to treat people effectively. So this is a map of some of the epidemiological studies done on cholangiocarcinoma. There's a lot of numbers on here, and you can ignore those. What I really want you to be looking at is which countries are colored in. So the countries that are colored in show where we have robust enough cholangiocarcinoma research to be able to publish on the epidemiology of it. And a large part of the world, majority of the world, isn't even represented in these studies. So that's a major limitation of cholangiocarcinoma research thus far. And this is one aspect that this project is trying to address. As I was talking about earlier, there are a lot of risk factors for cholangiocarcinoma, like liver fluke infection, inflammation, primary sclerosing cholangitis. But there's a lot we don't know that can fall into this environmental category, this inflammation category, and this host genetics category. So today we're going to be talking specifically about that genetics aspect. And I'm going to go into a little bit of what we're doing in this study with some of the scientific background that you'll need to know to understand the results that we're getting. So you have your genes, which you inherit half from your mother, half from your father, and we all have 23, not all of us, most of us have 23 pairs of chromosomes. So 23 is like the 23andMe company. And within those chromosomes, we have units called genes. So genes all have a specific protein that they code for. So each gene then has like a specific effect on the body. And within those genes, you have the DNA, which is all made up of A, T, Cs, and Gs, as shown at the bottom of this picture here. And an important term when looking at genetic studies is single nucleotide polymorphism, or SNP. I'm going to be referring to that a couple of times throughout this presentation. So if the letters that are crossed out here, the G and the C, those would be the DNA that a majority of the population has. And SNPs, this T and this A, are variants that a certain proportion of the population has, anywhere from between 50% all the way down to like 0.0001. So those SNPs are what we're going to be looking at these studies. So these are the most common types of genetic variants. I'm trying to use the word variant here instead of mutation. There's a general push in the scientific community to not use the word mutation because that has a pretty negative connotation, even though these single nucleotide polymorphism variants are very normal. So it occurs in about 1 out of 1,000 nucleotides, the component of DNA, and in any individual, there are probably 4 to 5 million SNPs. So calling them mutations is a little bit of a misnomer. They're the variants that naturally occur, and a vast majority are non-disease causing. However, if there's a variant within important parts of the genes or important parts that are between genes, it can affect the function of the protein. And that can have any sorts of downstream effects from that, including predisposing people to cancer. So in this study, we're going to specifically be looking at these single nucleotide polymorphisms that are predisposing people to cancer. And we're doing this through what's called a genome-wide association study, or GWAS. So this is a huge study that is sequencing DNA to look for these SNPs. In this study, we're sequencing large numbers of cases who are the cholangiocarcinoma patients and controls who are people without a cholangiocarcinoma diagnosis. And when I'm talking large, I'm talking on the order of thousands. We expect a lot of these variants to be very rare. And so just statistically speaking, if it's only present in 1% of the population, you'd need 100 people just to see it once. So we're looking for thousands of participants so that we can really see some of those rare variants that would be associated with cholangiocarcinoma. And so we're looking for those rare variants, and those would be less obvious in small populations. So that's the benefit of these GWAS studies. So we're going to talk now about what we understand about the genetics of cholangiocarcinoma currently. And when we're looking at genetics research, there are really two goals that researchers are keeping in mind, and two long-term outcomes that can come out of these genetic research studies. The first is understanding risk. So that's a huge one. I'm sure that as sequencing technology becomes more affordable and accessible, and insurance companies are covering it more, they're being taken into account in clinical settings more frequently. So somebody who has a family history of cancer will be able to go into the clinic, get their genes sequenced, and have a better understanding of their risk of developing cancer. So that's one outcome that we're really looking for when we're doing these genetic research studies. And the second outcome is developing targeted therapies. So if we know which gene is causing the problem, which is causing the cancer, we might be able to develop targeted therapies to prevent or treat that cancer. So what we know about cholangiocarcinoma genetics right now is that they're complicated. There are some cancers that we have very clear genetic risk factors for example, BRCA mutations in breast cancer. But for cholangiocarcinoma, there are no obvious genetic links. The inheritance within families on the population level is low. But we do see families that have more than one member with cholangiocarcinoma. So we can't say that there is no genetic link because we are seeing evidence of that. We just haven't been able to necessarily identify what that link is. A lot of the known genetic links can be attributed to general risk factors, which include primary sclerosing cholangitis, it's associated with ulcerative colitis. So those autoimmune diseases, a lot of genetic links can be through that, but we don't think that that's the whole picture, which is why we're doing this study. So variants, I had mutations in this PowerPoint. So variants found in cholangiocarcinoma are common cancer-causing mutations that are seen in many types of cancer, but we're interested in finding out what variants would be specific for cholangiocarcinoma. What we do know is that the genetic profile is different between different types of cholangiocarcinoma. So between intrahepatic and extrahepatic cholangiocarcinoma, we're seeing a different genetic profile. However, as a caveat to all of these things that we know, these studies have been really small numbers of patients involved. And that's because cholangiocarcinoma is a rare cancer and a single institution. And so usually research is done just at one institution, or at least that has previously been the model. A single institution probably isn't seeing large numbers of cholangiocarcinoma patients each year, which makes it hard to get those really big numbers needed to look for rare genetic variants. So this is one study that came out in 2012 that looked at genes that are associated with cholangiocarcinoma. So a big thing to know when looking at this data is that these are variants that are found in tumor samples, as opposed to variants in blood samples, and we would expect those to be different. So these are likely the variants that are causing the cancer. And a big thing to note from this is a lot of these variants, KRAS, TP53, APC, those are associated with cancer in general. Those are definitely not specific to cholangiocarcinoma. And another thing to note is that this study only contained 62 patients. So we can't draw large conclusions from that. One thing that they did note, again, is that there's a difference in the genetic profile of intrahepatic versus extrahepatic cholangiocarcinoma. So the dark blue is intrahepatic, the light blue is extrahepatic, and we are seeing differences between that. So the specific genes in this case, don't worry about it, but just note that we are seeing differences, and that's something important to keep in mind as we're doing genetic research on cholangiocarcinoma. So now that was all introduction. Now we're going to be going into what this project really is about. And the International Consortium for the Genetics of Biliary Tract Cancers. So this international consortium is an effort led by Dr. Lewis Roberts at the Mayo Clinic, who is my mentor, and Dr. Catherine McGlynn at the National Cancer Institute. And they are leading the first genome-wide association study of cholangiocarcinoma. So again, we're trying to get thousands of patients, thousands of people who don't have cholangiocarcinoma so that we can compare the two and look for those rare genetic variants that would be associated with cholangiocarcinoma development. This was split into two phases, the discovery phase, which happened around 2019, and then the expansion phase, which is happening right now. So in the discovery phase, there were 2,610 cholangiocarcinoma patients. And this is across 24 sites, all of which were in North America, Europe, and Australia. So this is also important to keep in mind because when we're thinking about studies that happen in North America and Europe, we're mostly thinking about people of European descent. There are a lot of white people that are involved in these genetic studies, and in this discovery phase, that's what we're seeing. And I'm going to talk a little bit more about that and why that's important. So this is a distribution of the patients that are in the discovery phase. Again, as I said, a vast majority of them, 2,400 out of the 2,900, are of European descent. We're seeing good representation from both intrahepatic and extrahepatic cholangiocarcinoma patients. About 200 of them were PSC-associated cholangiocarcinoma patients. And then there were limited numbers of people of Hispanic descent or unknown ethnicity. So in the analyses, for the sake of consistency, the discovery phase was all done in this cohort of European patients. So I'm going to go in and I'm going to show a little bit of the results that we found, but this is an example plot so that I can walk through what this study is doing. So we are drawing blood from patients with and without cholangiocarcinoma, and then we're sequencing the DNA. And the way we're looking at it is in this left panel here, these are patients without cholangiocarcinoma. These patients in red and the patients in green are patients with cholangiocarcinoma. And we're looking for those single nucleotide polymorphisms or SNPs, and how frequently they occur in patients without cholangiocarcinoma versus patients with cholangiocarcinoma. So in this example, the red variant here is what we can call normal, and the green one is the variant that would be that single nucleotide polymorphism. And so in this group of 3 of people without cholangiocarcinoma, we see it once, 1 in 3. But in the patients with cholangiocarcinoma, we're seeing it 2 in 3. So it has double the prevalence in the cholangiocarcinoma group. And then imagine that except on the scale of thousands of patients. So even though this is only a difference of 1 person, if we're sequencing hundreds to thousands of patients, we're going to be able to see more nuance between those 2 groups. And then when we're looking at how we identify which genes are important in our data, what we're doing is we're essentially dividing what we find in the cholangiocarcinoma patients with what we find in patients without cholangiocarcinoma. And we're making what is called a Manhattan plot here. So don't worry about the specifics, but essentially what it is the 23 chromosomes are all lined up, and the higher up it is on the chart, the more significant it is, the more likely statistically that it is a true result. And we put 2 lines on this chart. One is the line of statistical significance. So we run statistics on it and we can say there's more than a 95% chance that this is a true result. And then a line of clinical suspicion where it's like, well, these pink ones don't go above the statistically significant line, but we're definitely seeing a peak here. So we're suspicious of them. So now we're going to look at actual data. So in this European cohort, we're seeing peaks that come above that line of statistical significance. So this is showing us, hey, we do think that there's a real genetic difference between patients with cholangiocarcinoma and patients without. So the specific genes and points don't matter, but we are seeing points above the line of statistical significance and even more points above the line of clinical suspicion. And that clinical suspicion line, if you increase the number of patients that you include, we'll expect these dots to rise up potentially above this line of statistical significance. So this is only in 2,400 patients. And if we expanded that to 10,000, let's say, there's a decent chance that these middling dots will rise up above the red line. And this is a good example when we're only looking at the intrahepatic samples, 900 patients. We're not seeing any that rise above the line of statistical significance. So even cutting the study in half, we no longer see statistically significant samples, and we'd want more patients in order to see those, because we are suspicious of these ones. Likewise, with looking at patients with extrahepatic cholangiocarcinoma, specifically in males, this is only 600 cases. We see a lot of suspicious points, but nothing that we can say with confidence that this is a true variant. Dr. Young-Hun Han at Baylor did a big analysis on all of the things that we just saw. And this is new data. This is very exciting. And in all of that data from the 2,600 patients, he was able to estimate a heritability of 12%. So what that means is that genetics can account for 12% of the likelihood of getting cholangiocarcinoma. So we're not seeing, a huge 95% association, but 12% is still very real, and that it's very real for a lot of patients. So that's showing that the study of the genetics of cholangiocarcinoma is important. There probably are very true results that we'd then be able to translate to clinical practice eventually. And another thing that we saw is that the heritability is different between different types of cholangiocarcinoma. So between intrahepatic and extrahepatic cholangiocarcinoma, and between cases that are associated with primary sclerosing cholangitis and cases that aren't. So while they're all cholangiocarcinoma, we're really seeing differences between these different subgroups, which means that it's important to study those subgroups individually. Overall, we found 5 novel variants, 1 that's in all patients, 2 that we only saw in extrahepatic cholangiocarcinoma patients, 1 that's in PSC related, and 1 that's in non-PSC related cholangiocarcinoma. So we have these 5 novel variants, and we would expect that number again to increase if we have more patients involved in the study. For an example, 1 of the associated variants has previously been associated with cervical, kidney, prostate, and thyroid cancers. So there's a good chance we're on to something by associating this with cholangiocarcinoma as well. And a number of the variants found were within what's called HLA regions. So HLA regions are genes that are associated with your immune system. They're associated with immune responses and with autoimmune diseases. And when we think about the known risk factors for cholangiocarcinoma, and we think about primary sclerosing cholangitis, ulcerative colitis, Crohn's disease, those are all autoimmune diseases. So it makes sense that we're finding variants in the HLA regions because cholangiocarcinoma is associated with these autoimmune diseases. So now we're moving into the expansion phase of this study. We were recently awarded a grant by the National Institutes of Health to help fund increasing the number of patients in the samples. So we started with the 2,600 patients. We have a goal of sequencing an additional 7,000 patients over the course of the next 5 years. And the exciting thing about this is that we don't just want to look in North America and Europe as we did in the discovery phase. We want to really take this project globally. And right now we're working with over 82 sites from over 31 countries. And one of the groups we're working with is the Cholangiocarcinoma Foundation. And they've been a huge part of our ability to both fund this study and be able to recruit very generous patients to consent to being in this study themselves. And increasing the amount of patient samples that we have will allow us to look at even rarer variants. So like I said earlier, if a variant is in 1 in 100 patients, we'd need to sequence 100 in order to see it once. But there are some variants that are in 0.1% of the population. And so increasing the number of patients we sequence to closer to the order of 10,000 will allow us to see those even rarer variants. And like I pointed to earlier, there's a big emphasis in this project on global genetic diversity and increasing representation in scientific research in general. So the next 2 slides show just a very interim analysis that I ran on the patients we currently have. So all of these patients are through the Mayo Clinic, and that includes, though, patients through the Cholangiocarcinoma Foundation because samples are routed through the Mayo Clinic. And so this data includes 200 participants through the Cholangiocarcinoma Foundation. About half of those are cholangiocarcinoma patients themselves, and then about half of those are participants without cholangiocarcinoma. And so far, just through the Mayo Clinic, we're able to look at about 10,000 cholangiocarcinoma patients and about 450 control patients. And this is just basic demographic information that we have on the patients. And looking at this born in the US, almost everyone that the Mayo Clinic sees is born in the United States, which makes sense given its location. And so this is just a snapshot of what's going to be included in the final study. And like I said, it's important to consider intrahepatic and extrahepatic cholangiocarcinoma separately, since we do see differences in the genetic variations, and we are seeing good representation between intrahepatic and then perihilar and distal combined as extrahepatic. So hopefully we will be able to see those nuanced differences between different types of cholangiocarcinomas. And then when we look at some comorbidities among these patients, we're looking at their medical history. We're saying, what diagnoses do they also have? So in this interim analysis, we looked at peptic ulcer disease, primary sclerosing cholangitis, ulcerative colitis, Crohn's disease, and then lumping ulcerative colitis and Crohn's together to look at irritable bowel disease in general. So again, this is the about 1,000 cholangiocarcinoma patients, 450 control patients. And these numbers are the counts. So 101 out of the 1066 have peptic ulcer disease, which is 9.5%. So this is in cholangiocarcinoma patients and in controls. And this column here, the odds ratio, that's saying what are the odds that a patient with X, peptic ulcer disease, primary sclerosing cholangitis, et cetera, are also in the cholangiocarcinoma patient group. So all of these medical diseases will have positive associations with cholangiocarcinoma. For example, patients with peptic ulcer disease are at 4.2 higher odds of developing cholangiocarcinoma in this cohort than the control patients. And it's important to note in this cohort, so we only have the data on the patients we looked at. It can be different in other groups of patients, which is why we're trying to get as many patients as possible from as many places in the world as possible. So this is, again, looking just at Mayo Clinic and Cholangiocarcinoma Foundation patients, but we're seeing associations with peptic ulcer disease, primary sclerosing cholangitis, and irritable bowel disease. This last column here is showing how statistically significant these numbers are, and smaller p-values are better when it comes to statistical significance, and these are all e to the minus something, so they're all very, very small numbers, which is great. This is showing that there is likely a true association between these medical history and cholangiocarcinoma development, and this is replicating a lot of previously published results. This is just a subset of the data we're collecting. I'm collecting 164 data points. So each of these is one data point. We're collecting 164. So these are ones that have robust previous research behind them, but we're also looking at a couple of things that don't necessarily have as robust previous research. And so hopefully we'll be able to identify some new or suggested associations. And then our final plan is to associate this medical history information with the genetic information. So that's kind of the cool thing about this study, is that we're getting access to not only genetic information, but this clinical information. And we can run tests that test how strong the association is. For example, someone with ulcerative colitis might have a different genetic risk profile. So if someone has ulcerative colitis and variant X, they might be at the higher risk of cholangiocarcinoma. So this will all help us build, hopefully, a quite robust risk profile of the disease. And I want to talk a little bit more about understanding diversity in genetics. So all humans have a vast majority of their genes in common. But as we talked about earlier, each person has 4 to 5 million single nucleotide polymorphisms in their genes. So all of us have these natural genetic variants that get passed from parents to children. And because of that inheritance, we're seeing certain variants in different regions of the globe. in different proportions. So someone in Sweden might have a different distribution of those variants than somebody in Nigeria. And so it's important to have really diverse groups of patients in genetic studies. Vast majority of genetic studies that have been done globally have been done in North America and Europe. And so research participants have been overwhelmingly white. And it's great to have that information. That's really important to patients and to researchers and doctors to have that information. But it's also important to have information on people who are not white, because we might be seeing different risk profiles depending on where your genetic ancestry is. And so in this study, we're using what's called a global diversity array. So this is how we're sequencing the data. There are a lot of methods for sequencing DNA that look at variants that are found almost exclusively in patients of European ancestry. But this method of sequencing the DNA looks at variants found in diverse global populations. So it's looking at 4,800 genes and 1.8 million variants. So through this method, we're going to be able to collect a lot of information on not only patients that have previously been sequenced, but patients globally. And this map shows the countries that have sites that we're working with. So you can really see that global distribution. And I want you to think back to that first map that we saw, where a vast majority of the world wasn't represented. And this is showing how we're trying to branch out and really get better global representation in this study. And so where the Cholangiocarcinoma Foundation generously comes in is they are helping us recruit patients for this study. So there are ways that you could participate in the GWAS. I'm going to show two different ways. This first one is for people who are not attending the Cholangiocarcinoma Foundation conference next month in Salt Lake City. So we are able to ship supplies to you if you'd like to participate, even if whatever hospital you go to doesn't participate generally in this study, you as an individual can. So we're recruiting both cholangiocarcinoma patients and non-patients. So I know a lot of you are caregivers, family, friends. So anyone can participate, anyone over 18 can participate in this study. And so if you scan this QR code or go to the link that's written in the bottom right here, this is going to take you to the cholangiocarcinoma website where there's information on the study. So the way that it will work is that you can submit your contact information. You'll be contacted by someone in our team to consent you to the study, make sure you know what the study is, and make sure you consent to participate in it. And then you will receive a blood draw kit in the mail, and you can bring it to a clinic of your choice for blood draw, and we will be able to cover the costs of whatever that blood draw is. And then the blood can be shipped back to the Mayo Clinic where we'll sequence it. This remote process is being led by Nellie Campbell, who's a member of my lab and is doing an excellent job helping us out with this. So if you're not going to the Cholangiocarcinoma Foundation conference and you'd like to participate, this is the link for you. And as a side note, if you do scan this link, it might be a little bit until we contact you because we are going to be really busy with the Cholangiocarcinoma Foundation conference. So we promise we have your information, but it might take us about a month to actually get in contact with you after registering. And then this is for people who are going to be able to attend the Cholangiocarcinoma Foundation conference next month. This is just a little bit of information. So again, this is a research study of genetic variations. We take a small sample of blood for people with and without cancer, and we're looking at better strategy. We're looking to develop better strategies for prevention, screening, diagnosis, and treatment of cholangiocarcinoma. So this is the informational webinar. And we're also going to be giving a couple of updates at the conference itself. So I will be giving an update for patients and advocates on Wednesday, April 17th, and then another update for the scientific and medical community on Friday, the 19th. This is being sponsored by the National Cancer Institute, Mayo Clinic, and the Cholangiocarcinoma Foundation. So we really appreciate them for that. And this is the QR code if you would like to participate through the conference. So we are going to be there at the conference with phlebotomists and with our team to consent patients. We're going to have a room where the blood draws are happening, so you can consent now and then get your blood drawn at the conference, and it'll all be put together hopefully smoothly so that you can participate through your participation at the conference. So this is the QR code for you if you are going. And finally, I'd like to thank everyone who is involved in this project. It's a huge project, so there's work from a huge number of people. Dr. Roberts is my mentor, and everyone in the lab, especially Nellie and Matt, are part of this. We have a consenting team led by Dr. Fawzia Ahmed and several people in the lab. The consortium leadership, Dr. Catherine McGlynn, Dr. Amos, Dr. Hassan, and Dr. Young Han Han. We'd like to thank the Cholangiocarcinoma Foundation for their funding and support. I'm funded through the Mayo Clinic MSCP program, and this project is funded by the National Cancer Institute. And this is a list of the sites that are participating, and they all deserve acknowledgement as well. So now we're moving into a question and answer session, and this is my email if you have specific questions that you'd like to send me after the webinar.
Perfect. That was great, Caitlin. Thank you for that great explanation. It made a very difficult concept easy to understand. I appreciate that. If you want to leave your contact information up there for now, that would be great, because I think I saw some of those were the questions in the chat. And for everyone, please put your questions in the Q&A box or the chat box. Please try to keep them general questions about the GWAS study, not personal questions, as Caitlin won't be able to answer any personal medical questions. Okay, so how do we connect with you? That your email address would be the best way.
Mm-hmm.
My husband had cholangiocarcinoma, as did his brother. His sister had liver cancer. I have 2 daughters, so they could all potentially participate. Yes?
Yes, absolutely. An important thing to note is that due to the ethical nature or the ethical concerns involved in genetic research, at this stage in the research, genetic information will not be relayed back to you. So this is purely for research and discovery at this point. Once the findings are developed and we have good evidence, there is a possibility of changing the way that this information is collected and routed. And so in the future, there's a potential of being able to access the information that through this research study, but at this point, you will not be able to access the genetic information that we get.
Thank you for pointing that out.
On an individual level. This will be available through presentations, through publications, potentially through news articles. So you'll be able to see the results of the study, just not your individual.
Won't receive any personal results back to you, exactly. But you are contributing to this very important research, which is very much needed. Exactly. Okay. Are the NRAS and KRAS the same mutation or variant, I guess you would like us to say?
Those are different genes, and they're both genes that are involved in cancer development generally, and there are a lot of different variants within those genes that lead to cancer development. So just because those genes make a certain protein, and that protein is important in preventing cancer, when we see variants in those genes, there's a higher likelihood of someone developing cancer.
Thank you. Have you excluded Lynch syndrome families from your study?
We haven't. We're taking all cholangiocarcinoma patients. There are some more nuanced requirements for control patients where we're sequencing everybody who can be consented, but in certain analyses, we might exclude patients with other cancer diagnoses. So I know families with Lynch syndrome tend to have a lot of cancer in them. So in certain parts of the analysis, we might not be able to use that information.
Okay. What kind of genetic testing should the family members of a cholangiocarcinoma patient be getting with that 12% risk factor?
Yeah, that's a great question. And currently we don't know. That's really what this study is trying to figure out. There's not a genetic panel in the way that there are for some other cancers like breast cancer, colon cancer. Currently, there's so little known about the genetics of cholangiocarcinoma that we're not sure what to look at. I know that in patients with multiple family members with the same disease, or in this case, the same cancer, you can work with medical geneticists at your hospital to try to figure out a personalized approach, but generally there's not currently anything.
Okay. Can you comment on the correlations between cholangiocarcinoma and Lynch syndrome and or having a mismatch repair inherited gene?
Yeah, so that mismatch repair is one of those genes that helps prevent cancer. So mismatch repair genes, when there is damage that happens to DNA, mismatch repair genes go in and they fix that damage. And if you don't have mismatch repair genes that are functional, so if those have a variant in them that makes it non-functional, you're not able to go in and fix those that damage that happens. And so you're at risk for a lot of different types of cancers generally. I don't know the specifics on the overlap between Lynch syndrome and cholangiocarcinoma, but damage and mismatch repair genes can make it more likely that you develop any kind of cancer.
Okay. So I think the answer to this is no. Would non-patients receive their genetic testing results?
Correct, no.
Wouldn't collecting related family members as controls dilute any possible association signal? Unrelated spouses might be a better choice.
That's a great question. So that is something that's considered when doing these big analyses. For some of them, we do exclude first-degree relatives, especially. So first-degree relatives being your patients or your parents, your siblings, your children. Controls do function better if they aren't related, except it is important to look at those familial links as well. So looking within a certain group of related individuals, are there variants that we're seeing among those related individuals? Or variants that we're seeing in the cholangiocarcinoma patient, but not in their sister or their mom or their kid.
So you still encourage all of those people to participate, whether they're related closely or not?
Absolutely, yeah.
Okay. Under suggested associations, have you considered type 1 diabetes, which is the result of a few autoimmune diseases? A further subset that anecdotal evidence of people on the CCF Facebook groups would be adult onset type 1 diabetes. Have you seen anything to suggest this association?
Yeah, absolutely. So we're seeing association with a lot of autoimmune diseases, including there are suggestions that there's an association with type 1 diabetes. We are collecting that information. I hadn't thought about the adult onset aspect of that. That's interesting that you bring that up, and that sounds worth looking into on my part. So thank you for that suggestion.
Awesome, thank you. Okay, again, the answer no on this. You will not receive information back on genetic variants.
Correct.
If I had cholangiocarcinoma, but am currently no evidence of disease, would I be participating as a cancer patient or a non-cancer patient?
Good question. So we're looking at DNA from the blood, which is the DNA that you're born with. So regardless of whether you are cancer-free now, congratulations, you would still be counted as a cholangiocarcinoma patient for this study.
All right, a lot of thank yous for the great presentation. And would you put up the QR code for the non-conference patients one more time?
Absolutely.
And no, you will not receive genetic counseling after if you participate. This one, you will not get the results returned to you at this time. Is there any use for genetic results of my husband and brother who passed away already? Like, is there any opportunity for deceased members if they have those results?
That's a good question. And I see another question in the chat about previous genetic testing. So the way that people sequence DNA between research projects, between companies is different. So prior genetic testing information would be harder to incorporate into our results. So that's not something we're looking at this point in time. If whatever hospital or clinic that they went to has blood in storage in like a biorepository, it would potentially well, I'm gonna say no, sorry. I think that like the ethics around that would be hard to work through.
Okay, good try though.
Yeah.
If I'm being seen at one of the sites that the study is being done, are you already in it, or do you have to ask about being a part of it?
Yeah. So we're working with a lot of researchers at a lot of different sites, as you saw on the list on the map. Each of them has their own research study that they're collecting patients through. So it's possible, but not guaranteed that you're already involved. I think you'd have to talk to your doctor about it.
Be safe to ask. Yeah, okay. So again, another mother had passed away and had gene testing done. Are those results at all helpful to your study?
Not at this point.
Are Canadian residents included in your blood draw program?
Yeah, we have a lot of international participants. We have a partner in Toronto right now, the UHN, UNH. We don't have a lot of sites throughout Canada. And as far as participating in this method where we ship you the kit, we'd have to look at shipping costs. I think that it would come down to logistics. So you can contact us and we can look into it, but I'm not sure about the finances of it.
But if you're a Canadian patient at the annual conference, you can participate.
Absolutely, absolutely.
Okay. PSC and UC are kind of old news as risk factors. Can you please highlight other areas of interest you have been seeing, such as smoking, obesity, drug use, environmental exposures, anything along that line?
Yeah, definitely. Those are very well established risk factors. I'm still collecting that information. So I have 164 different things that I'm looking at, and the things that you listed are involved in that. But I don't have that information to share yet.
Okay. Are you using artificial intelligence to partner to assist with the research?
Yeah, that's also a good question. I'm using it like informally to help me with things. But there is a possibility of using all of this information and training artificial intelligence to develop a prediction model. So we're not at the point where we have enough information to go through that, but it's definitely a possibility in the future.
Okay. So along the lines of the conference, should they wait till they get there to sign up to participate in the study, or would it be better if they consented earlier with that QR code you gave them?
It would be better to consent earlier. It will be possible to consent at the conference, but the more we can get done ahead of time, the less frantically busy we'll be during the conference.
Yes, exactly. We'll put that QR code out and hopefully send it to everybody who's registered at the conference, so you guys will have an opportunity to pre-consent and get as much done ahead of time. Okay, so I think we kind of covered this earlier, but would you recommend children of patients with cholangiocarcinoma have genetic testing done now?
Not at this point, unless there's like specific genetic variants that you know about your parent. I think you'd have to work with a medical geneticist about that.
Okay. And when, do you have a timeline of when we might learn more about the genetic variants that you discover?
So the first set of variants from the discovery phase that I presented on a little bit here, we're working on putting together a manuscript for that. That will hopefully be published within the next couple of months. So that initial data set, hopefully very soon. This expansion phase, probably about in 4 years.
Okay, and a suggestion, Lynch families are at increased risk for cholangiocarcinoma, so I think it should be exclusionary or at least subset out of the rest of the pool. And then, do you know if FGFR2 fusion mutation can be genetic?
I do not.
Yes,
Someone's
and
on
no,
no,
we can't just send in reports. You definitely would have to join with a blood draw. Again, asking if EGFR3 is a genetic factor and are there any studies relating to it, medications for it? Are these slides going to be available to share?
I know it. Can I get back
We
to
have the recording, but if you can't share the slide, yeah, we'll have the recording available on the YouTube channel that we can share. And if Caitlin finds out she can share the slides, we'll be able to do that as well. Yes, we'll send the recording. Sorry. How can people limit damage to mismatch repair genes? Are there links to diet or sleep patterns?
Interesting. So this isn't my area of expertise. The variants that you're born with, you can't control. But there are things that you can do. For example, wearing sunscreen is a big one for your skin cells. Things that are associated with cancer in general, like smoking, can help limit additional damage.
Okay, and again, to emphasize for international patients, they can participate at the studies internationally that are doing it, but right now not necessarily able to participate for you guys sending the kits out internationally. But if you're attending the annual conference, international patients can do it there. Okay. What if international patients are willing to pay for the cost of the shipment of the kit?
We need to look at what that cost would be.
Case by case situation.
Depending on the way we ship it, it can be upwards of $1,000. So we'd have to look at that particular situation.
Nice offer.
Yeah.
Okay. If you have received a blood draw kit, how long do you have to return your sample?
I don't think that there's too much of a limit on it. I don't know how long the tubes are good for, but I think quite a while. Once the blood is gone, limited amount of time, but just sitting with the kit in the box, you have more flexibility.
Yes, but once you get the blood drawn, I think there's definitely a time limit that's explained in there to get it shipped right away. Okay. How can we stay informed about your study?
Good question. So I have been in touch a lot with the Cholangiocarcinoma Foundation. I gave a similar webinar last year, and hope to be invited back in the future when we have some more results. Like I said, we'll be at the conference. So our team in general keeps in really close contact with CCF. So hopefully you should be able to see things through them.
Would non-patients have a strong family history of CCA be eligible for GWAS? Would they be in the control group? Yes.
Yes.
Okay, yes, we will share the recording. It will be up, and I will see what we can do about emailing all the registrants the both codes to enter into the study, whether you're coming to the conference or not. Do you know if the Cleveland Clinic is participating?
Yes. I don't know the name of the specific researcher though, off the top of my head.
Okay. Do you know if there's any link between celiac disease and cholangiocarcinoma?
Good question. So celiac is one of those autoimmune diseases. I don't know specific numbers, but yes.
Okay.
And that's one of the pieces of information we're looking into.
Okay. If you have another cancer besides cholangiocarcinoma as well, can you participate in the GWAS study?
You can participate, and we would want to know that.
Do you know if there's any sites in Chicago?
Yes.
WashU, St. Louis.
Northwestern, yes, WashU. We
Okay.
have worked with University of Calgary.
And maybe we can get that list of participants from you and send that as well.
Yeah, that's a good thought.
And post it. Okay. And the one from Canada as well. Oh, actually, the international sites would be great.
Mhm. I can send a list of all sites and contacts there.
Okay. Yep. Most of the other questions are just asking what sites are particularly participating. MSK, Johns Hopkins, but we'll get that list and we'll see if we can provide that information to everyone. And the one from Canada as well. Okay.
Absolutely.
I think we have answered all the questions. Thank you so much. We look forward to the patients who are coming to the annual conference in a couple weeks. Hope you guys can pre-consent. We'll get this information out to you so you can get started on it. And those who are not, please use this QR code to get signed up on the website and get the process started. We appreciate all the work you're doing, Caitlin, and the Mayo Clinic and Dr. Roberts and everyone else who's a part of this. And we look forward to seeing you in a couple weeks and getting a lot more samples for you.
Great. Thank you so much. And thank you everyone for coming.
Yes. Thanks, everyone. Have a wonderful day.
Bye.
Bye-bye.